All right. So thank you, Professor. Thank you, Sai, for this this opportunity. And today I'll be presenting for you guys some preliminary data that I have about my research, uh, which has the title of seasonal variability of enzyme activity under different cropping systems. So as an introduction, we know that crop productivity per unit area here in Southern Great Plains is lagging, and it's due by several problems, but one of these problems is related to soil health, and it leads us to the need of more intense and diversified cropping systems in order to improve not only just nitrogen and water use efficiency, but also soil health. And talking about soil health, I have this diagram. It's simple, but it just explains that soil health is divided in chemical, physical, and biological properties that are affected by several factors. Some of them are listed in the picture, as you can see: land use management, the soil type, the cropping system that you are adopting in your farm, in the farmers, the inputs, and the climate, sorry. And so all these factors influence these three parts of the soil that together will be describing soil health. So the objectives for this study is to assess the dynamics of enzyme activity during the winter crop growing season and how the cropping systems through intensification and diversification will affect these enzyme activities. And we hypothesized that the best time for soil sampling for biological indicators is on fall or with winter wheat planting and emergence, and that enzyme activity has a positive correlation with other soil health indicators. So here we know that this intensification and diversification is important to enhance, maintain soil health, but we will also need to know when is the best time to collect the soil to measure the soil health. So the rain plots, just a second. Going here, I think that. Did you guys see any picture, or it's just stopped? Okay, here we go. Okay, so yes, the rain plots are located in National Bottoms, Riley County, Kansas. So we have Wilmore Silt clay loam soil. The experimental design is our CBD with 4 applications, and we assessed the soil in 3 depths: 0 to 5, 5 to 15, and 15 to 30 centimeters. So here is just a diagram of all the treatments that we have. So you can see the treatment number 1, which is a control. It's just a sequence of winter wheat and summer fallow. And you guys can see on your right side, we have the name of the treatment, and right underneath that, we have the intensity index and diversity index for each one of the treatments. So treatment 2, we have a sequence of winter for hay and for soybeans with 1 season of summer fallow. We have treatment number 7, a semi-intensive with a sequence of wheat and soybean with some summer fallow as well in the middle. For the intensive treatment, we have a sequence of winter wheat, sorghum, summer fallow, soybeans, winter wheat, sorghum, triticale, corn, and triticale. For treatment number 12, it's a dual purpose semi-intensive. We have winter wheat, summer fallow, triticale for hay, soybeans, winter wheat again, a summer fallow, triticale for hay, a summer fallow again, and followed by triticale for hay. And treatment number 17 is a dual purpose intensive with winter wheat, moth, dual purpose wheat, alfalfa, triticale, alfalfa again, dual purpose wheat, alfalfa, and again, dual purpose wheat. So we can see that we have a wide range of intensity indexes as well as diversity indexes. And talking about these indexes, intensity index is just the rate of crops in a rotation divided by the years, and the diversity, it will consider the different kinds of different type of plants, which could be grasses or broadleaves in your system. So we have like a wide range of intensity and diversity through these 6 cropping systems. So going a little bit about material methods, continuing, this is like a big picture about what I am measuring at field level. So I will be measuring biological properties as well as chemical and physical properties. But for today, I will be focusing only in these 4 enzymes that were measured by the colorimetric method with our dried soil samples. So my statistical analysis, I used the mixed model with the specific enzymes activities as response variables. I analyzed each one of the enzymes by depth with treatment crop stage interactions as explanatory variables and the blocks as random variables. After that, I ran an ANOVA and followed by a pairwise comparison. So going through the results and discussion, you guys can see a table for each one of the depths, where at the topsoil 0 to 5 centimeters, we have significance for the cropping systems and the growth stages, and for the other 2 soil layers, 5 to 15 and 15 to 30, we have less significance for the cropping systems and more significance in between all the enzymes for growth stage from the culture. Talking about beta G activity, we have for the first layer, 0 to 5, the letters indicate the mean difference between crop stages. We can see a clear difference or a decrease in activity in the blooming stage on the first soil layer. For the second layer, 5 to 15, we can see pretty much the same pattern. And for the third layer, 15 to 30 centimeters, we have a cropping system effect, and you can see a different pattern where we started in the planting with lower activity. We have a boost on activity on the blooming stage and kind of decrease for pretty much all the treatments going through the harvest point. One thing that I want to highlight is that the presentation will follow the colors. So red is topsoil, green is the middle layer, and the intermediate layer, and blue color is the last layer for all the plots. Going to NAG activity, responsible for nitrogen cycling and also carbon cycling. I forgot to mention that beta G is also responsible for carbon cycling. We have in the first layer significance for treatment and cropping stage. We can see that it doesn't have the same pattern as we saw on beta G, having kind of a decreased pattern going through blooming and through the harvest point for pretty much all the treatments. When you go to the intermediate layer, 5 to 15 centimeters, we have an effect only for growing stage, having a decrease from planting to harvest. And for the last layer, 15 to 30 centimeters, we have a boost, a really expressive boost on activity on the blooming stage, having even higher values than topsoil in the blooming stage, and also going to decrease through harvest. Talking about acid P, really important for phosphorus cycle in the soil. We can see an effect for treatment in our cropping system in all the growing season. So we can see that acid P has a more stable behavior or more stable dynamics grow along the growing season in the 0 to 5 centimeters layer of soil. We can see a decrease from blooming towards to harvest in activity in the intermediate layer, and also a decrease pretty much. We have kind of boost also in blooming stage, but going to a decrease in the harvest stage. Going to aerosulfatase activity. Stop again. Just take some time. Here we go. So for the first layer, we see for some treatments an increase on activity during the blooming stage, and going back down or lower activity going through harvest stage on the first soil layer. In the second one, we can see almost the same behavior, having in some treatments some boost on potential activity on blooming stage, but really going to lower activities to the harvest stage. And on the last soil layer, 15 to 30, we can see also that some treatments just decrease activity from planting to harvest. Some of them just reached lower levels at blooming and just maintained the activity levels until crop harvest. And here on this plot, I just want to highlight that we have on the x-axis pretty much all the growing season, talking about months, and then in one of the y-axis we have precipitation, the bars, and mean temperature, the line. And the three lines represent gravimetric water content on the soil or soil moisture, and you can see like a really decrease in water content in the soil, even though we had some big rainfall events, as you guys can see in the bars, but we also had high temperatures, high mean temperatures, and also we have crops growing in the plots. So with high water uptake, which decreased a lot on available water or soil water for the soil microbes and enzymes activities. Here, it's a correlation plot, and one of the hypotheses we hypothesized that some enzymes would have good correlation with other soil health indicators, or in this case, organic matter. And you can see that 3 of the 4 enzymes, aerosulfatase, acid P, and beta G, have a good correlation with soil organic carbon as well. Yeah, my PowerPoint's not working so good today. Okay, and then I also calculated beta G and ag and beta G and acid P ratio to identify some nutrient limitations So in beta G neg ratio, looking for nitrogen limitations, and the ratio between beta G and acid P, looking for phosphorus limitations. So I calculated by dividing the natural log of beta G by the natural log of neg, and the same thing for acid P as well. So values smaller than 1 for the ratio beta G neg would represent nitrogen limitations, and values greater than 1 would represent carbon limitations. It's the same thing for acid P. So values smaller than 1 would represent phosphorus limitations. So going through beta G neg ratio, we can see that we have more carbon limitations in topsoil going towards harvest point, and more nitrogen limitations on the last soil layer, and more representative on the planting stage and blooming stage for some treatments. For BG acid P ratio, we can see observed in all the treatment depths and crop stages being more expressive on the last layer during planting stage, so more phosphorus limitations. Here, I try to explain like how much each factor would explain the variability of my data. So I run a mixed model with all the variables as random effects. And we can see that for all the enzymes, depth is really important. And we have some effect of treatments and crop stages as well. So just like kind of descriptive plot to see, okay, how much each one of the variables will explain or explained the variability of my data. So my conclusions are that we had different dynamics through the growing season between the 4 enzymes. We had, like, for beta G, a decrease in activity during antithesis. We have, for example, for neg, a decline in activity during all season, but that boost on 15 to 30 centimeters. For isophthase, we also had a decrease in activity during antithesis, and for acid P, we have a more stable dynamic along the season. And this decrease in potential activity for some enzymes in all or each one or all the growing seasons can be related for environmental factors and plant-soil interactions. We know that the enzymes and biological factors from the soil are really sensitive to environmental factors. And then the optimal moment for sampling for biological measurements on crops emergence, but it will depend on environmental factors in the next growing season that will be analyzed. And also that we had a good correlation between 3 enzymes and soil organic carbon, so acid P, isophthase, and beta G. So the next steps, we'll be adding the microbial community analysis to this research. They are being analyzed already. We are analyzing and sampling, analyzing this summer crop season that's going now. So we'll have data for soybean, corn, and 2 other summer crops as well. We'll have a second year repetition for winter crops. So the next winter wheat crop will be analyzed as well in order to create more reliable data, and also take a deep look on the relations plant-soil that can explain this dynamic of the enzyme activities along the growing seasons. And that's it. Thank you all for your attention, and I am open to questions.